presentation today about the rat experimental model for investigation of the effect of diabetes on submandibular salivary gland treated with epidermal growth factor. As an introduction, animal model provide a unique opportunity for researchers to study the development of the disease and their complication under controlled conditions that limits the influence of the genetic and environmental factors on the progression of the disease, including diabetes. In diabetes research, different animal models have been developed and used to study type 1 diabetes by spontaneously developing an autoimmune disease or ablation of the beta cells of the pancreas. While in type 2 diabetes, animal models have different degrees of obesity, insulin resistance, and pancreatic beta cell failure. Diabetes mellitus, also known simply as diabetes, is a group of metabolic diseases in which there are high blood sugar levels over a prolonged period of time. It's characterized by presence of hyperglycemia, glucosuria, and polyuria. This results in elevated blood glucose levels, which leads to a number of complications, including nephropathy, neuropathy, cardiomyopathy, retinopathy, atherosclerosis, and diabetic foot ulcers. Moreover, diabetes is associated with increased susceptibility to bacterial infection, inflammation, caries, and xerostomia, and periodontal diseases, and also related to with a malfunctioning of the salivary gland and salivary flow. Investigation demonstrated that insulin and diabetes affect the function and structure of rat parotid gland found after 1 month of diabetes induction in rats. There are inhibition of the gland growth in diabetic rats. By histological examination of the gland, there were an excessive accumulation of lipid droplets within the acinar cells cytoplasm together with increase of lysosomal activity. Moreover, it was found that the dense vacuoles and crystalloids were found in the apical cytoplasm of the parotid gland of striated duct cells. Epidermal growth factor is a powerful protein that has a significant role in wound healing through facilitating the proliferation and migration of fibroblasts. Epidermal growth factor identified while the studying nerve growth factor in submaxillary glands of male mice. Epidermal growth factor is known to be produced in human salivary gland, increase the protein synthesis and glycolysis, as well as increase in gene expression and eventually increase in DNA synthesis and proliferation. Myosin include a family of an ATP-dependent motor proteins, are best known from the role of muscle contractions and their involvement in a wide range of other eukaryotic motility processes. They are responsible for actin-based motility. The term was originally used to describe a group of similar ATPs found in striated and smooth muscles. The aim of this study. The aim of the present study was to validate the diabetes mellitus and epidermal growth factor experimental rat model and to detect if the epidermal growth factor could reduce a well-known side effect of diabetes mellitus induced by streptozotocin on submandibular salivary gland on adult male albino rats or not. This was verified through immunohistochemical detection of myosin in parenchymal end of the gland. Material analysis. Animals and housing 80 animal albino rat, male albino rat, adult age 3 months, average 220 grams. Albino rats divided into 4 groups. Control group was injected the same number of saline injection as epidermal growth factors. Diabetic group, single intraperitoneal injection of 65 milligram per kilogram of freshly prepared streptozotocin dissolved in zero point 0.1 milli sodium citrate buffer. Epidermal growth factor, a single injection of epidermal growth factor intraperitoneally in a daily dose for 2 months. Last group, diabetic and epidermal growth factor. Rats were treated with the streptozotocin, then injected the single daily injections of epidermal growth factor. Myosin immunohistochemical detection of the myosin was performed by in glandular tissue using a staining reaction induced by anti-myosin antibody. Imaging of the all specimen and photographed by Zeiss microscope with a 10 megapixel camera. Statistical analysis of the myosin expression was performed in terms of mean standard deviation analysis of the variance of ANOVA test. Results. Histological results of all groups, including control group, with a negative staining of the cytoplasm of the mucus secretory cells, was evident in the epidermal growth factor group, while negative to weakly positive staining of the cytoplasm opposite to the lumen and intercellular canaliculi of the serous acini was observed in the same group. Strongly positive staining reaction was found around nearly all the secretory cells and intercalated duct representing the staining of myoepithelial cells. Intercalated ducts and striated ducts, granular convoluted tubules, and excretory ducts demonstrated weakly positive stains. In diabetic group, the negative stain is weakly positive stain of the serous secretory cells. The myoepithelial cells surrounding the periphery of the acini and the intercalated duct showed weakly positive stains, reaction in the cell bodies, and weaker stain reactions in the cell processes. The intercalated, striated, and granular convoluted ducts reveal that negative to weakly positive stain. While in epidermal growth factor group, negative stain of the cytoplasm of the mucus secretory cells was evident, while negative to weakly positive staining of the cytoplasm opposite to the lumen and intercellular canaliculi of the serous cells was observed, with strongly positive staining reaction were found around the secretory cells and intercalated duct of myoepithelial cells. The diabetic group treated with epidermal growth factor, examination of the sections taken from the diabetic group treated with epidermal growth factor are including anti-myosin antibody revealed obvious improvement in comparison with those of diabetic group. Negative staining of the cytoplasm of the mucus secretory cells, while negative to weakly positive staining reactions in the cytoplasm was evident opposite to the lumen and the intercellular canaliculi of the serous cells. The myoepithelial cells were elongated with bodies and the spider-like extension of their process shows a strongly positive staining reaction around and nearly the secretory cells and the intercalated duct. The intercalated duct, striated, granular convoluted tubule, and the excretory duct reveals weakly positive staining reaction. The statistical analysis revealed histomorphological analysis of the submandibular salivary gland with 3 groups revealed that the highest mean area percent occupied by myosin immunostaining was recorded in the control group, whereas the lowest value recorded in the diabetic group. ANOVA test revealed statistically significant difference in the mean area percentage of expression of myosin immunostaining reaction. A comparison was held between the myosin mean area expression percentage in the control group and other experimental groups using independent t-tests. Statistically significant decrease in the myosin expression area percentage in diabetic group and diabetic treated with epidermal growth factor group when compared with control group. There was no statistically significant difference between myosin expression in the control and epidermal growth factor group. Furthermore, there was a statistically significant decrease in the myosin expression area percentage in diabetic group treating with epidermal growth factor when compared with control group. Finally, there was a significant increase in myosin expression area of percentage in the epidermal growth factor treated and a diabetic group treated with epidermal growth factors when compared with the diabetic group. In a conclusion, the experimental rat model described in the current study is a simple, reliable, and predictable animal model. It can be used in diabetes and epidermal growth factor research. The epidermal growth factor administration in a daily dose of 10 microgram per kilogram body weight for 2 months intraperitoneally leads to enhancement of all the submandibular salivary gland component. This was proven by immunohistochemical localization of myosin. The finding from the present study confirm previous results from the studies using scanning electron microscopy, cytokeratin expression, and the same animal model. Thank you.